So we're going to continue on the theme of the tree of life, starting with looking at the relationships among the three domains and the implications of the particular tree or trees that we infer on understanding the biology of different organisms, and then talking a little bit about, if we have time, about some complications in the tree of life. I'm going to probably change the order from doing viruses right at the end. All right, so they claim that clickers work now in here. We'll see. If we don't detect all the signal, we won't count it. All right. So given these, this tree, the three-domain tree, where we have a polytomy in the tree, how many different ways are there to resolve this tree? Again, I'll give you to about a minute and a half. All right, last chance. All right, 63% said B. Just going to try and get this out of the way. And the correct answer is B. Congratulations to those 63%. So this is analogous to the polytomy I showed earlier with humans, chimps, and gorillas. And for this particular polytomy, it's 3 branches coming out of this node here, and we have to resolve the order among those of those branches. Here's one possibility with bacteria and archaea as sister groups. Here's another possibility with bacteria and eukaryotes as sister groups. And here's a third possibility with archaea and eukaryotes as sister groups. So those are the 3 ways you can resolve this sort of tree of life, 3-domain tree polytomy. And that's, again, equivalent to this polytomy that we saw before. We had a 3-organism polytomy, humans, as I point to chimps, humans, chimps, and gorilla. And there were 3 ways to resolve that. Is that any questions about this? All right. So you may remember when we talked about the human, chimp, gorilla resolution, one of the ways to resolve the relationships among these taxa was to have an outgroup. And with parsimony, character state reconstruction, at least the method we use, we can use the outgroup to help us determine what the ancestral character states are for a particular group, and then that helps us infer what the relationships are among the taxa. So what is the outgroup here? This is all of life, all modern taxa in the tree of life that at least Woese could characterize at the time. So this is the problem with this, with resolving this tree, is that this is the outgroup, basically. or nothing is the outgroup, nothing that we have modern data from in terms of taxa. So remember what an outgroup is. An outgroup is a lineage that's separated prior to the existence of the common ancestor of the in-group. And if all of life is our in-group, we need some lineage that's separated out prior to the existence of the common ancestor of all of life. And unless there's a fourth branch on the tree of life hiding out there somewhere, we don't have that in modern organisms. So we need some alternative to root the tree of life, and that's why Woese, when he created a tree, and when many people still draw a tree based upon ribosomal RNA data, they show it as an unrooted tree, or a tree with a polytomy like this. And so we're not going to go into a lot of detail on why a particular rooting is shown in the class, but in the next few minutes I'm going to explain to you one of the lines of evidence for the particular rooting that's used for the tree in the class. And it's a really interesting, incredibly creative idea that a few scientists had in the late 1980s, early 1990s. And what they did was they thought about the evolution of all the different modern taxa. And if you draw the phylogenetic tree of these, I've drawn these really thick bars because we're going to imagine evolution of genes inside the genomes of these taxa. And so this is sort of showing the core pipe of evolution of the entire genome of these taxa. And then we're going to imagine what happened with the evolution of individual genes inside of their genomes. And if all was simple, which of course it is not, but if all was simple, if you looked at the evolution of an individual gene within any of these, within these lineages, it should follow the same phylogenetic tree as the rest of the genes, or of the whole genome, or of the species, basically. So the gene should have, eukaryotes should be related to each other, archaea, bacteria, and you'd have the same polytomy if you used ribosomal RNA or any other gene. So another gene might look like this, another gene might look like this, and so on. And what they observed and then figured out how it would be useful is that it turns out prior to the existence of the common ancestor of all of life, inside the genome of that lineage, of that organism, some individual genes that were present made copies of themselves, a duplication of that gene. And then inside that organism's genome, those two different duplicates started to evolve separately from each other. And so inside the genome, there were now these two distinct lineages of duplicated genes that were evolving separately. There's a special term for those. They're called paralogs. We'll come back to that later. You don't need to know that right now. But those two distinct duplicates evolve in parallel within the genome of each of these individual taxa. And what they realized was that if you look and you compare version 1 of each of these genes to each other, the tree should look just like these trees. But if you build an evolutionary tree that includes version 2 of those genes at the same time as version 1, version 2 of the gene can serve as an outgroup for studying the evolution of version 1. And so I'm just going to sort of show you a diagram of what this might look like. One of the genes that people did this with is a gene for what are called elongation factors. They're involved in the translation of RNA into proteins. And if you look across all taxa, every organism has two different forms of elongation factor of a gene. Those different forms are related to each other. They are homologous, but they are distinct. And the only plausible explanation for these two different forms existing in all taxa is that some ancient event, a duplication happened where there was the proto-elongation factor gene, and then a copy was made of that gene inside the genome of the common ancestor of all of these organisms. And so you can imagine the gene evolution undergoing a split, where there was this ancestral single elongation factor and then a split into two distinct lineages inside the genome of this common ancestor. And then those evolve separately in parallel. And when there are speciation events, when the species split, each of the duplicates now track with each of those descendant lineages, like I've shown with the purple for what's called elongation factor TU, one of the elongation factors in all taxa, and elongation factor G is shown in blue. So when you have the lineages split, each of those descendants gets a version of these genes. And what you can do is if you build an evolutionary tree of elongation factor G, and you want to, in essence, root that tree, and you want to use, say, the outgroup of that tree to infer what the pattern of character states was, the ancestral state. For the tree of elongation factor G, you can use any of the elongation factor TUs, EFTU, as the outgroup. So if you made a big alignment, a big data matrix, you could have EFGs in the data matrix, and I've just abbreviated them with a 2 here in the data matrix. They are related to each other, but all of the EFTUs are more closely related to each other than any is to EFG and vice versa. So if you consider this your in-group for your tree, you could use these as your outgroup for the tree, the EF2s for the EFG and vice versa. No, this may sort of be a new type of thinking for everyone, but that evolution of different genes within a genome is a common occurrence. We have it all the time in our recent ancestry. So all the different globin genes that animals have, so you may be familiar with alpha globin and beta globin and gamma globin and a lot of different globin genes that that code for proteins that become subunits of hemoglobin in our blood. Those originated by duplications in the past history of vertebrates. This is the same concept, but just a very, very, very long time ago. So when you take that approach and you build evolutionary trees with EFG or EFTU, what people found, again, in the late 1980s, early 1990s, was that Archaea were a sister group to Eukaryotes. And that's why in the book and in many other discussions, the tree that is shown for the tree of life has Archaea and Eukaryotes as sister taxa. Are there any questions about this duplication analysis to find outgroups? Yeah. It's what? It's not based upon the character per se, it's based upon the sequence inside the elongation factor, the genes for elongation factors. And if you build a tree of either of them and then root it with their duplicate, you get a tree that looks like this. Now, I'll come back later in the lecture to talking about how this tree, we do not see this tree for every gene and every genome, but it is the reason why in the book and in many other parts of the literature, this tree has been used for a long time as the rooted tree of life. There is some evidence for that rooting of the tree. Does that answer your question? Okay. Any other? So first of all, one complication that we'll come back to later is that even though this was the rooted tree found by those duplicated genes in the 1980s and 1990s, and even with some analyses of duplicated genes today, depending on which phylogenetic reconstruction method you use and which data sets you include, so now there's a lot more data than there was 20 or 30 years ago. We have complete genomes from across the diversity of life, or across much of the diversity of life. There's an alternative tree that many people get when they build trees of all of these lineages, which has the organisms that are called Archaea right now being polyphyletic. And eukaryotes actually, in essence, evolving from within the Archaea as a lineage rather than as a separate group from Archaea. It's unclear right now which of these two trees, this one with 3 domains, each being monophyletic, or this one with eukaryotes evolving from within one of the branches of Archaea are the best tree to represent the data. And as we'll see at the end of the class, there may be a better explanation for this if we don't think of the tree of life as a single tree, but I'm going to come back to that later in lecture. Oh, sorry, this has been known as the eocyte tree. You'll see this sometimes in the literature. It was named that by the first person who found this tree. So, but for now and throughout the book, we're going to treat this tree as the winning rooted tree of life. So now what we're going to do is walk through how we can use this rooted tree of life to better understand some of the features that are present in these different groups. And in particular, we're going to start with features that are, in essence, widespread within one of the domains, but different either in the other two or in where many or all of the taxa in one of the groups has a feature that's different than in the other two groups. Here's another clicker question. Given this rooted tree of life, prokaryotes can be described as paraphyletic, monophyletic, quasiphyletic, and multiphyletic. I'll give you to 1:15 this time. Everybody ready? All right, good. It was hard not to laugh when I read out quasiphyletic, but the correct answer is A. And I'm just going to show you what that looks like. Prokaryotes are paraphyletic. That's what paraphyly looks like. Archaea and bacteria are both prokaryotes. If this rooted tree of life is correct, that means that, as I sort of suggested in the lead-in to talking about Wosa's tree, that the distinction of prokaryotes and eukaryotes was, in essence, a misleading parameter in terms of inferring the relationships among taxa. Prokaryotes are not monophyletic according to this rooted tree. So again, what I'm going to do is walk through a couple of examples now of features of different taxonomic groups, the major groups, and show you how we can use that rooted tree to learn a little bit about the history of those features. So for example, this is now starting to get into chapter 26 a little bit as we're now going to shift from chapter 22 into chapter 26 and chapter 27. So one of the sort of standard examples used to discuss the three branches on the tree of life is that the membranes in Archaea, or at least in many Archaea, are relatively unique. They have a special chemical linkage. You don't have to know the chemical structure here, called an ether linkage. And the membranes, again, surrounding the cell of bacteria and eukaryotes are ester linkages. So given this particular pattern of membranes, we can try and reconstruct the evolutionary history of when these features originated. One possibility, which is the most parsimonious possibility, is that the ancestor had ester linkages, and then in the branch leading up to the common ancestor of Archaea, there was a switch to ether linkages. Yeah? Did I get it backwards again? Yes, sorry about that. These are reversed. This is an ether and that's an ester. And she just told me this before class too, and I got it wrong again. That's one example of using the tree to track evolution of features. There are many features that may be unique to some of these branches. So here's an example with unique bacterial features. So here's an example of a unique bacterial feature, which is peptidoglycan. Peptidoglycan is a polymer of sugar, these amino sugars, that's found in the cell wall of bacteria. There are actually two different kinds of bacterial cell envelopes. We'll talk about this later, gram-negative and gram-positive. In both of them, though, the bacteria use peptidoglycan as a material in the outside of their cell. It's called the cell wall. And this particular chemical is not found outside of bacteria. And so if you overlay this onto the tree, here bacteria has peptidoglycan, Archaea and eukaryotes don't have peptidoglycan. Again, we can use the rooted tree to infer something about the ancestry. So we're going to do another quick clicker question here. Using parsimony character state reconstruction, what can you infer about the ancestral character state for this group of organisms in terms of peptidoglycan? Okay, till 1:20 here. Anyone need extra time? All right, till 1:30 then. Ready now? All right, the majority, 57%, said C, which is the correct answer. And so let's look at the reconstructions here. Here's one of the possible reconstructions with peptidoglycan being the ancestral character state and then a change on the branch leading up to the common ancestor of Archaea and eukaryotes, or alternatively no peptidoglycan and then a transition on the branch leading up to the common ancestor of bacteria. So actually this rooted tree does influence what we infer about the properties of what some people call LUCA, the last universal common ancestor, or sometimes referred to as MERCA, the most recent common ancestor. So having a rooted tree influences what we infer about or can't infer about the properties of the common ancestor of all of life. And we're not going to talk about that really much in this class, but a lot of people are interested in what the common ancestor was like, both because they may be looking at fossil information or ancient rocks to try and look for evidence for the earliest life forms, or they may be looking at how life may have evolved. And if you want to model how life evolved, it's very helpful to know what the first life forms were like, what their features were, and this type of reconstruction is necessary to do that. Okay, so now let's move on to where there's lots more stuff going on. Which is unique eukaryotic features. This is the hallmark feature of eukaryotes. Not the only one we'll talk about, but sort of the key defining feature. The term eukaryote basically means present true nucleus, contains a nucleus. So the nucleus is this membrane that surrounds the DNA in eukaryotes. Does all sorts of important functions, but it's a compartmentalization of the genome away from the rest of the cell, from the cytoplasm and other components. And if we now look at the nucleus as a trait and overlay it onto the tree, gotta get rid of my little clicker thing. What we would infer is that the origin of the nucleus happened sometime on the branch leading up to the common ancestor of eukaryotes. That's the most parsimonious explanation, at least, given this rooted tree. And it turns out there are dozens to hundreds of other major features that are shared by eukaryotes and different or absent from bacteria and archaea. And clearly, something major happened on the branch leading up to the common ancestor of eukaryotes. For example, it's not just that the DNA is contained within the nucleus, but that containing of DNA has allowed for many different forms of regulation and processing of RNA compared to bacteria and archaea. So, for example, DNA is transcribed into RNA in all taxa, but in eukaryotes that's done in the nucleus, and then the RNA is exported from the nucleus out to the cytoplasm where translation happens. And because of that process, or related to that process, eukaryotes are able to do many other functions that we don't see in bacteria and archaea. For example, in many eukaryotes, the coding regions of genes that code for the final protein are not contained within a contiguous part of the DNA of the genome. The cell will transcribe a very large RNA, export, and then cut out parts of that RNA in a process called splicing before generating the final RNA that is then used to make proteins. And that process is enabled by having this separation where transcription happens inside the nucleus and translation happens only when the RNA gets outside of the nucleus. And this splicing allows for all sorts of complexities of creation of proteins. So one gene in eukaryotes can have, the separations are called introns and exons. One gene can have dozens of exons. Those can get differentially spliced to different proteins. So one individual gene can be made into thousands, if not hundreds of thousands of different proteins because of this process of splicing. But yeah, is that a question or just yawning and stretching? You should be familiar with many other unique features of eukaryotic processes. So eukaryotes copy themselves in asexual reproduction by a process called mitosis. Eukaryotes undergo meiosis when they're producing gametes to undergo sexual reproduction. Eukaryotes, many of them have linear chromosomes. At the end of the linear chromosomes, there are special chromosome caps called telomeres that are involved in the maintenance of the length of the chromosomes in eukaryotes. And the counter is also true. There are many sort of processes that if you look at how they work in bacteria and archaea, they're similar between bacteria and archaea and different in eukaryotes. So asexual reproduction in bacteria and archaea happens by a process called binary fission, where the cell basically duplicates its chromosome and then splits itself in half. We will talk later about gene transfer mechanisms where DNA is exchanged between different prokaryotes, and that is in many ways similar to sexual reproduction, but it's has different effects and different processes. And so in general, what we see is an enormous number of events happened on the branch leading up to the common ancestor of eukaryotes that gives them features that we don't see in bacteria and archaea, or features that are different character states in bacteria and archaea. That basically, everyone with me here? So again, the prokaryotes, this sort of simple feature that was used to divide up life into two major groups, is not evolutionarily informative. So what I want to talk about now is a little bit more about just what exactly happened here in the origin of eukaryotes. And I switched the order a little bit, so we're going to come to viruses later. This is a little different order than in the slides, and talk about what I call complications, but also really focusing on eukaryotic evolution first. So if we, this goes back to a question that was asked before about elongation factors. If we look in the genome of any organism, but let's just start with eukaryotes. If we look inside the genome of one of these organisms, the DNA in eukaryotes is packaged most of the time into chromosomes, and the DNA is sort of wound up into layers and layers of these bundles of coils. And when an organism needs to make something off of part of that DNA, it gets unwound and opened up, and then the transcription process happens to make RNA off of the DNA. And anywhere along these threads, these can be, in humans, some of the 23 chromosomes are, tens to hundreds of millions of bases in length, an incredibly long string of DNA. And contained within that string are thousands of genes. And if you look within the genome, when I showed you the WOS tree of life and even the elongation factor tree, it's based upon a tiny, tiny fraction of the total genome of any particular taxon. And it turns out that it's a good marker of evolution. If we scan through the genome of a eukaryote and we build evolutionary trees of different genes in the genome, many of them look like this. Many of them, all the eukaryotes, share a common ancestry with each other, and if we can root them, many of them look like eukaryotes and Archaea as sister taxa to the exclusion of bacteria. And we can scan through the genes and they look something like this. But maybe surprisingly to some, if you haven't heard this before, certainly surprising to many biologists, when people started looking in detail at the different components of the eukaryotic cell, they found that many of the components of the cell and many of the genes in the genome had a phylogeny that looked like this instead, where eukaryotes were actually, that part of the genome in essence was contained in a tree within the bacterial branch of the tree of life. That is, eukaryotes, that part of eukaryotes was more closely related to bacteria, to some bacteria, than they were to other bacteria or to Archaea. And the converse, some bacteria for that part of their corresponding part of their genome were more closely related to eukaryotes than they were to other bacteria and Archaea. This is pretty unusual. And if we go back to the genome of a eukaryote and we scan through it and we look for genes for which we can compare across all these three branches in the tree of life. So there are many eukaryotic genes that are only seen in eukaryotes. We can't build a sort of big comparative tree of those because there's nothing to compare to in Archaea and bacteria. But for the ones that we can compare, many of them look like the three-domain tree, as I was mentioning before. Ribosomal RNA and a diversity of other genes look like this when we build trees. But a reasonable fraction, a smaller fraction, but a reasonable fraction look like this tree, with eukaryotes, again, nestled in the middle of bacteria. And so this basically means that eukaryotes, you can't draw a single tree to represent the evolutionary history of eukaryotes. And in fact, since eukaryotes are part of the tree of life, we can't draw a single tree of life that only has one branching pattern. There are multiple trees for different parts of the cell, different parts of eukaryotes. Any questions about this? Okay, so one of the explanations for this comes to back to the cell biology of eukaryotes. And so if you look inside a eukaryotic cell, you find all sorts of complex compartments, complex features. You have the nucleus surrounding where the DNA is kept. You have the endoplasmic reticulum and the Golgi apparatus and all these other things you should have learned about at some point. And you have this really interesting other part of eukaryotic cells called the mitochondria, and it's where cellular energy is generated in many taxa. And if you look at this mitochondrion with a good microscope, you will see that there are many similarities, physical similarities of the mitochondrion to bacteria. This was observed before anybody knew or the proposal that Archaea existed, so people focus primarily on bacteria. But since bacteria and Archaea look a lot like each other, at least much of the time, the mitochondrion also can resemble some Archaea. But it certainly looks like there's another cell inside of a eukaryotic cell. And because the exact appearance of a bacteria or an archaean is not a useful indicator of what their evolutionary history is, the exact explanation for this bacteria-like organelle inside eukaryotic cells took a little while to come to a full understanding of what was there. It turns out that mitochondria have their own DNA, and you can pull out that DNA, read the sequence of the DNA. It turns out that they have ribosomal RNA genes. So you can read the sequence just like Woese did of those ribosomal RNA genes and build a phylogenetic tree of those ribosomal RNA genes. And when you do this, it's very clear that mitochondria are derived in evolutionary history from bacteria. And in the, I think, 1960s, Lynn Margulis came up with a model to explain the history, what the mitochondrion. Other people had just sort of viewed it as yet another thing inside the eukaryotic cell. She was the person who really popularized the idea that mitochondria were derived from bacteria. There had been actually a literature from the early 1900s in Russia that had also talked about this, but people had forgotten it, and she rediscovered this notion, and she proposed this model, which many people thought was silly, ludicrous, ridiculous at the time. It turns out to be dead on correct, which was, imagine that there was an ancestor of all eukaryotes. And this ancestor reflects the part of the phylogenetic tree that shows eukaryotes grouping with Archaea. And so maybe this was an Archaea-like cell, maybe it was a little different, the branch separated off from Archaea, and maybe it became a little bit different. It's unclear if it had a nucleus then or if it evolved a nucleus later. Let's propose that it didn't have a nucleus yet, but it has its own genome and its own cell membrane. And this organism evolved symbiosis, an interaction with a bacterium. And it's a particular type of bacteria called proteobacteria. We'll come back to that in the next few lectures. You don't need to worry about that now. But this bacterium had its cell envelope, including peptidoglycan, and actually two layers of a membrane, one inside the peptidoglycan, one outside the peptidoglycan. The primitive eukaryotic, sort of Archaea-like ancestor engulfed this bacterial cell, brought it inside of its own cell membrane. And when you do this, when an organism feeds, many sort of protists and amoeba and other cells or white blood cells will envelop particles. When they do this, the particle gets the cell membrane from its host wrapped around it. Lynn Margulis had noted that mitochondria look something like this, with a cell membrane around them from the normal host cell, in essence, and then bacterial-like membranes, at least, around the mitochondrion. And then, at some point, the DNA of the host cell was enveloped into a membrane to create the nucleus. Again, that may have occurred first or later. It's not exactly clear. And over evolutionary time, eventually, what was a potentially free-living bacterium lost part of its biology, part of its functions, and became an obligate, dependent symbiont that lived inside the cell of other organisms, and that this was the ancestor of all eukaryotes. So that means that the different parts of the genome of this cell. There's the part that shares ancestry with Archaea, and there's the part that shares ancestry with this one lineage of bacteria, the Proteobacteria. So again, this would explain why some of the genes inside eukaryotes, if you build trees, look like this, and why some of the genes look like this. Eukaryotes, again, are chimeras. They have multiple evolutionary histories within the individual cell, and that this was the proto-eukaryotic ancestor. Are any questions? Yeah? Yeah, so the question is there an example for the tree of life of representation with more than one tree? What we're going to see as we go along, and I'll show you some examples of this, is people try to represent the phylogeny of life as a network with some crossing branches rather than as a single bifurcating tree. Because it turns out that lineages merge after they separate occasionally. And so you can't perfectly draw this as a bifurcating tree. We still use the term tree of life to sometimes generally refer to the evolutionary history of life, even though there's really no way to represent it as a single bifurcating tree. That answer your question? Any other? Yeah. So the question is, did the mitochondria lose its peptidoglycan? I didn't say this when I showed this, but I'll show you. So the model is that the ancestor of the mitochondrion originally had all of the cell envelope of a proteobacteria, which would include an outer membrane, then a peptidoglycan, and then the inner membrane. And no modern mitochondria have peptidoglycan. So the explanation is that it lost the peptidoglycan. It turns out there are a few weird parasitic bacteria that also have lost their peptidoglycan. So it's not really possible to distinguish from this simple model whether or not the original symbiont had already lost its peptidoglycan or if that happened after. But there are no modern mitochondria that have peptidoglycan anymore. Okay, so one more complexity in this story, which is that the genome of modern mitochondria is very, very small compared to the genome of most bacteria. And modern mitochondria don't carry out all the functions that a free-living bacteria does because the host does a lot for the mitochondrion. But there were many, many more genes at the beginning of this symbiosis than now. And another sort of amazing part of this story is that if you look in the eukaryotic nuclear genome that I was talking about, some of those genes in the nuclear genome have an evolutionary ancestry that they share with these proteobacteria. That is, those genes in the nucleus came from the mitochondria. And what's happened over evolutionary time is that DNA moves from the mitochondrial genome into the nuclear genome and eventually gets lost from the mitochondrial genome and kept in the nuclear genome and transmitted to offspring. So when eukaryotic cells divide, their offspring get mitochondria. The mitochondria make copies of themselves. They make copies of their genome. The offspring cells get a copy of the mitochondrion and a copy of the DNA in the nucleus. But occasionally, DNA moves from the mitochondrion to the nucleus. And over long evolutionary periods, that DNA has eventually stayed around in the nucleus and been lost from the mitochondrion. And what's really interesting is that many of those pieces of DNA code for proteins that still function in the mitochondrion. They now have to get transcribed in the nucleus, exported, translated, and transported into the mitochondrion to function there. But they very clearly came from evolutionary history shared with the mitochondria rather than the rest of the nucleus. So again, inside the nucleus, there are genes with different history, and two of sort of the big bands of history are some genes, eukaryotes share a common ancestry slightly more with Archaea, and some genes, they share this common ancestry with the ancestor of the mitochondrion. Are there any other questions about the mitochondrial story? We're going to come back to this when we talk about chloroplast evolution and early eukaryotic evolution, because chloroplasts are also derived from a symbiotic event very similar to this, but only in one branch of eukaryotes rather than a common ancestor of all of them. So in the last few minutes here, I want to talk about one other, you could say, complication that we left off the discussion of the tree of life, which is viruses. Viruses are incredibly diverse. Viruses are basically a particle made up of protein usually, and inside that particle is a genome, genetic material. But you can have viruses, some viruses have RNA as their genetic material, some have DNA as their genetic material. Those that have RNA or DNA, it can come as single-stranded or double-stranded. There's a lot of diversity in terms of what is in the genome, and there's lots and lots of diversity of the protein coat in the viruses and the functions that viruses carry out. But all viruses that have been studied are parasites of cells. So they're not considered per se true free-living organisms. But they do have genomes. Those genomes evolve. And one question is, where did viruses come from? How do they fit in with the tree of life? And one of the challenges with viruses is, although we can crack open cells and extract their ribosomes and look at their ribosomal RNA genes and build a tree based upon ribosomal RNA, viruses don't have ribosomal RNA. So when Woese was building the original tree of life in the 1970s, and even up until now, until people started to sequence large amounts of genomic material, the connections between viruses and cellular organisms was very poorly understood. It's still not particularly well understood. But basically, there are three models to explain the evolutionary history of viruses. I'm just going to show you those models here. So one of the models is that viruses represent, in essence, the fourth domain, a fourth major branch on the tree of life that's separated prior to the existence of the common ancestor of all bacteria, archaea, and eukaryotes. And that maybe in these early evolving systems, viruses, could have been carrying out certain functions. They're now obligate parasites, but maybe in the past they weren't obligate parasites, and maybe they're so weird in the sense they don't have ribosomal RNA, et cetera, because they represent a fourth major lineage in the tree of life. There are many papers that claim this. There are some viruses that have all sorts of features in their genome that people didn't used to ascribe to viruses. So there are viruses that have been recently discovered that are bigger physically than many bacterial cells. There's even a virus that has its own viruses that infects it. And there are viruses that have translation apparatus. So viruses are much more diverse and much closer to cellular organisms than used to be appreciated. An alternative model is that viruses represent a completely separate origin of sort of genetic material from primitive prebiotic systems with no sharing of common ancestry with bacteria, archaea, and eukaryotes. And then a third model is that viruses represent, in essence, escaped parasites. There are many parasitic bacteria and eukaryotes that infect cells of other organisms, and many of those parasitic cells have thrown away a lot of their cell envelopes, a lot of their cellular functions. They depend upon their host for all sorts of activities. Maybe one of those lineages just got rid of even its own cell membrane and only has a protein coat and now exists as what we call viruses. So these are three, there are other models. These are three models to explain where and how and if we can connect viruses to the tree of life. It's probably a little bit of each of these. There are examples of viruses that appear to be escaped particles from cells. There are examples of viruses that appear to be, fourth domain-like, very ancient evolutionary lineages. And so viruses are strange. They're really interesting, and we will talk about them a little bit on and off in the rest of the course. And we're going to continue on with more complications in the next class.